Okay, Vanessa, can you hear me?
Yes, I can hear you.
Okay. We're going to go ahead and get started. We got Zoom bombed on a couple.
Yeah, I was trying to stop on the videos.
Yeah, we'll have to fix that for the next meeting. I am going to be removing them as I see them. Sorry about that, everybody. But welcome, everybody, and welcome to the very first NextGen Epilepsy seminar. And we're very glad that you chose to join us today. I see people from all over the world, including some Zoom bombers here that we'll try to remove as we can. We may have to fix this for the next Zoom seminar series. So this is a series that's going to feature 12 really outstanding postdocs and graduate students who are going to present their work in epilepsy through research seminars all throughout the year. The speakers come from all over the United States, and I'm hoping this is going to be a good way for us to showcase some of the research that's going on in the field. So these are some of our very best trainees that we have in epilepsy research, and the talks are going to be moderated by myself and Momoko Watanabe, who is a new faculty member here in the Anatomy and Neurobiology department at UCI. She just joined us from UCLA, and we're hosting this series with the UCI Epicenter. And so before we start, I've asked Tally Barham to say a few words about the center and what we're trying to do here in epilepsy. Tally, are you here? I'm going to unmute you.
Mm-hmm. Can you turn my video on too?
Yep. Oh, you want me to?
Yep, I can't. You're in charge, and that will give you time to go get rid of all the wonderful.
Yeah, sorry about that.
No problem at all. So can you turn my video on?
I'm looking.
Sounds great.
I have to ask your permission.
Okay, I got you. You've got permission.
Cool, we're good.
Fantastic. This is really, really cool. I mean, regardless of the bombers, we have really a full room. And I think this is going to be terrific. I am really honored that Bobby has made, together with Momoko, have made this possible. In fact, what I think I would like to share with you is really what the Epicenter is doing now. The Epilepsy Research Center is part of UCI, is embedded in anatomy and neurobiology. And pandemic or not, we've gotten some pretty interesting milestones. Momoko's just joined, so is Laura Ewell, who's on here, and Javier Diaz-Alonso. And pandemic or not, we have new people coming in. Bobby has achieved serious milestones. First of all, he got his tenure, and also became one of the executive committee of the UCI Epicenter, which is really being rejuvenated, as you can see here today, dances notwithstanding. So we really are focused on epilepsy, but epilepsy is a window into brain plasticity, and I think this series is an epitome of that. Just to tell you a tiny bit, the Epicenter was really created, about 18 years ago. And for the women in the audience, it's been created by a team led by women and by a woman. And at that time, it was kind of unusual. Now it's fantastic and taken for granted. We've always had seminars. And if you want to go onto the website, you'll see that in addition to the NextGen, there's also another seminar series. There is a training program that just has a position open for those who are interested in joining us. And there are a number of activities. We'll have a symposium, hopefully non-virtual, in March. But fundamentally, we're here to promote epilepsy in any way, shape, or form, and particularly epilepsy research by people who are next gen. So I am like thrilled that we're having the series. So Bobby and Momoko, kudos. And I'm going to shut up and let you guys start. And you know where to find me, anyone, if you have any questions or anything you want to chat about.
Cool. Okay, thanks, Tally. That was great. And so now it's my pleasure to introduce our speaker today, Vanessa Nieto Estevez. So Vanessa is a postdoc with Jenny Shea. She's been there with Jenny since 2015. She got her bachelor's degree in biology at University of Alcalá. Did I say that correctly?
Yeah.
Oh, cool. And PhD in neuroscience at the Cajal Institute in Madrid. And so she's going to talk to us about 3D human brain organoids and epilepsy. specifically, I think, ARX mutations. And because of the, I'm managing the message board here. I've closed messages to everybody. What we'll do is save your questions till the end, and then we'll invite everybody to unmute and talk about and ask questions if they'd like to. Okay, so with that, Vanessa, you are on.
Okay. So thank you, Bobby, for the invitation. I'm really happy to share my work here today. And it's an honor to be the first one to open this series. So as you say, I'm going to talk about the project that I'm doing in the lab about epilepsy related with ARX mutation. I don't need to say here that epilepsy is a group of disorders characterized by recurrent seizures due to neuronal hyperactivity. It's a really common disorder, and there are many causes that lead to epilepsy. But in the lab, we are working in genetic epilepsy. And in many of the cases that the cause is a gene or a mutation in a gene, and the onset happened really early. So that means that sometimes even during development, you can see the phenotype. And in the last 20 years or so, more and more genes can be found mutating in patients with epilepsy. But in the lab, we are working, or we are interested in ARX. ARX is the aristallate-related omega box and is a transcription factor located in the chromosome X. For this reason, most of the patients are males. This is a schematic of the gene, and one characteristic of this gene is that it contains 4 polyalanine tracks, here in blue, and it depends on what kind of mutation and where the mutation are located. One of each is asterisk is one type of mutation. The phenotype could be more severe or less severe. So you can see in this picture, in the mutation that's produced to KIT protein is the more severe phenotype. And you can see here how in the patient with a mutation, they have this smooth brain if you compare it with the normal brain. However, the other type of mutation, insertion or mismutation, don't produce malformation. And this mutation include the polyalanine expansion, that is the one that I'm going to talk today. What is known about ARX is described in mice, and the first role for ARX was that is an important transcription factor for interneuron development. It's important because it can be a repressor or an activator or other transcription factor. And as you know, the interneurons are produced in the ganglionic eminence, and they migrate to the cortex, from there to the cortex. So for this reason, when you model this type of polyalanine expansion in mice, you can see a reduction in GABAergic positive cell in the cortex of the mice. In addition to that role of ARX, there have been shown also in mice using a knockout specifically in the cortical area and EMX1 mice. So using that mice, they also found that there are defects in the layer of the cortex. So you can see in this picture, they found a decrease in superficial layer here, STAT-B2 and COS1, but they didn't found different in deep layer neuron. So this defect was found in embryo, but is also maintained postnatally. And all of these defects promote the development of seizure in mice. However, we all know that mouse and human, we are different. And you can see here how the brain is different. And if we go to a cellular level, the main difference between mouse and human is that human contain, or they have this motor subventricular zone that is important for the expansion of the cerebral cortex in human. So, and also other difference between human and mouse, as I mentioned before, the interneuron in mice, they are produced in the ganglionic eminence and they migrate to the cortex. In human, in addition to that path, some interneuron can be also produced locally in the cortex. So for this reason, we decided to study the role of ARX using human cells. So what we did was recruit control and patient and we reprogram the blood to get a pluripotent stem cell. And with this cell, we differentiate them to organoids to have epilepsy in this model. So what we did, because ARX, as I mentioned, is important both in the cortex and in the ganglionic eminence, we differentiate the iPS cell to two different types of organs. The cortical spheroid, that they will be more similar to the cortex, and the subpallian spheroid, that they will be more similar to the ganglionic eminence. So you can see here, this is the culture condition that we are using. Basically, we are using the method described by Pascal. And for the cortex, we use a combination of factors. And for the subpallia, in addition to those factors, we add two more factors to ventralize the cortex or the organoid. So you can see in this picture, in the cortical spheroid, we can find ventricular zone-like structure where the progenitor cells are located around and the neuron outside this area. So we can also see a little bit later that we can start to see a deeper layer neuron, and even later we can see also upper layer neuron. And what is also important that in this organoid we can find the other radial glial cell, the cell that I mentioned that they are specific in humans. So you can see now in this picture, in this cartoon, that the human organoid, they are a little bit more similar compared with mouse to the real human brain. It's not exactly the same, but now we have the other radial glial cell and more expanded the layer of the cortex. The other type of organoid, the subpallian spheroid, I told you that these one are like the ganglionic eminence. So here we can see that they are enriched in GABA positive cell, and we can even see different subtype of GABA or the different subtype of interneuron like the calretinin cells. So now we have a model that we can use to study epilepsy, but the first question that we asked was what we will see in this model. So as I say at the beginning, if epilepsy is due to neuronal hyperactivity, we were thinking where or what can cause this neuronal hyperactivity. So we were thinking that could be due to defect in progenitor cell, an imbalance between interneuron and glutamatergic neuron, maybe migration defect, morphological changes, synaptic defect, or maybe in electrophysiological changes. So some of these aspects have been shown in mice, but we want to see what's happening in organoid or in human cells. So what we are doing here is just compare healthy iPS cell with patient. And as I told you, the one that we are analyzing is specifically polyalanine expansion in the second track of ARX. So the patient that we recruit, they contain 8 extra alanine, and we had also another patient with 11 extra alanine, but this one we couldn't include it in the study because we couldn't get good iPS cell from this patient. So I just showing here today the last three patients. So you can see even though the mutation is the same, the phenotype is slightly different between the patients. And in addition to the patient, we also have 3 control, and you can see here this they have just 12 alanine. So this is the characterization of the iPS cell. The first thing that we checked was that in fact, we had a pluripotent stem cell. So you can see here labeled with different pluripotent markers. And we also checked that the karyotype was normal after the reprogramming. So then we differentiate the iPS cell to cortical and subpallian spheroid. And in the first set of experiment, what we did was characterize both type of spheroid using immunostaining and single cell RNA-seq. So the first question, when we have all the organoids that we asked, was if ARX is expressed in this model. So you can see here are single cell. All the data that I'm going to present about single cell, I'm doing that in collaboration with another postdoc in the lab, Parul Ranma. So you can see here that when Parul analyzed the organoid, she found a cell that expressed ARX, both in the cortex and in the subpallian. And there are different cell types that express ARX. For example, in the cortex, we identified radial glial cell, other radial glial cell, progenitor, and also cortical neuron. In the subpallium, we also found progenitor and interneuron that express ARX, and as you can see in the number of ARX or cell expressing ARX is 10 times higher in the subpallium than in the cortex. So now that we know that ARX is expressed, we wanted to know if the expression is different when we compare a control and ARX or cell-front ARX patient. So you can see here, and we can expect from the single cell data that in the cortex that are this image, there are just a few ARX positive cells. And when we did the quantification, we didn't see difference between control and ARX. However, when we immunostain the subpallium spheroid, you can see here that now we can detect more positive cells. And at 60 days, we found a significant increase of the ARX expression in the ARX subpallium spheroid. So now we know that ARX is expressed, so we want to see what is the phenotype that this mutation is doing during development. So first we analyze the cortical spheroid. So you can see some example here, how the spheroid look like at different time points. And we found that at the beginning, at early time point, 14 days and 21 days, there is a slight increase in the size of the organ in the ARX condition. However, at later time point, the size is similar. So then we wanted to know what can cause this increase in the size of the organoids. So for this reason, we analyzed the proliferation of the cell. We used Ki67, and we found a significant increase of Ki67 positive cell in the ARX organ. Then we also want to know if different cell types are affected to this mutation. So the first cell that we analyzed was PAX6 positive cell. This one are progenitor cell. And you can see here how these PAX6 positive cells are located around this ventricular zone-like structure in both conditions. However, in the ARX condition, we found it's not significant yet, but it's really close. There is a decrease in the number of PAX6 positive cells in the ARX organ. In addition to that, we also wanted to label different type of cortical neuron. So we use CETIP, that is a marker of deep layer neuron. And you can see that now the pattern of where are located the CETIP2 positive cell is opposite to the PAX6. So now these CETIP positive cells are located outside the tubular region. And what we found here is that in the ARX condition, there is a tendency to have more CETIP positive cell in the ARX condition. So it's not significant yet, but it's close. So now by single cell, we wanted to see if we can corroborate this phenotype. So you can see here as the same that I showed you by immunostaining, we found by single cell. So we have an increase in Ki67 positive cell in the patient. We have also decrease in PAX6 positive cell, and we have an increase in deep layer neuron, as you can see here. Not only CETIP positive cell, but also CETP2. And in addition to that, we also found a decrease in upper layer neuron. So now we wanted to know if these effects are maintained over time. So now we have the same organoid, the cortical spheroid, but 120 days old. And now what we found is that instead of a decrease in PAX6 positive cell, now we have an increase. And also is opposite to the earlier time point, we have now a decrease in deeper layer neuron, but an increase in upper layer neuron. So then the next question was if these cellular defects are or have a consequence in the functional property of the organ. So that we did it in collaboration with another postdoc in the lab, Saint Laurent, and we used a multi-electrode array MEA to measure the spike and bursts in the organ. So you see here, what we have is a well that contains 12 electrodes. One of each line is the information from one electrode, the same here in each square. So you can see that we can detect the activity in each electrode. So what Zane did was analyze the number of spikes and also the number of bursts. And it's not significant, but you can see that there is a trend, that there is a decrease in the activity of the ARX organelle, both analyzing the spike and the burst. So just to summarize this first part, what I told you is that at the beginning, what is happening in the cortex is that there is a decrease in Paxis positive cell. And at the beginning, the neurons that are being forming are the deep layer. So what we think that is happening is that ARX is promoting the differentiation now of the deep layer neuron, that is the one that are being forming. However, at later time point, now there is a wave of differentiation of the upper layer neuron. So this is why the phenotype is opposite in a later time point. So now we have an increase in the Paxis positive cell, and now ARX is promoting the differentiation to upper layer neuron. So now I'm going to talk about the other type of organoid, the subpallian sphere. So again, the first thing that we did was analyze the size of the organoids. So you can see now that just by eye, you can see that in the subpallian, the phenotype is more severe or is more obvious. So you can see that at all the time point that we analyze, there is an increase in the size of the organoid. So again, the first thing that we did, because we found this increased size, we checked the proliferation of the cell. So we used Ki67, and now that is surprising, we found a decrease in Ki67 positive cell. We also use a marker of a progenitor of interneuron, NKX 2.1, and we didn't see any defect or any difference between ARX and control. At later time point, we wanted to see if there is an effect in apoptotic cell, and we use AC3, apoptotic caspase-3, activated caspase-3, and we found that there is an increase in AC3 positive cell in the ARX patient. In the same way that I mentioned for the cortex, Parol is doing single cell both at 30 days and 120 days. So here I just want to show you the UMAP. And now Parol, as we speak, she is trying to characterize the different cluster and all of that. This is 30 days. And this is 120 days. So again, Parol is trying to identify all the cell population. So the next set of experiments that I want to show today, so because we know that the brain is not just one area isolated to the other one, so we wanted to know what happened now if we fuse cortical and subpallian sphere. Because as I mentioned, the interneuron migrates from the ganglionic eminence, in this case the subpallian, to the cortex. So we want to analyze this kind of migration also in vitro. So to do that, the first thing that we did was label the interneuron progenitor using a DLX 1.2 GFP reporter. So you can see that in this way we can label many cells in the subpallian sphere, express this reporter. So then when the virus is expressed, now we fuse one subpallian sphere with one cortical sphere. So you can see here how the cells are labeled, and we just put them together. After a few days, you can see how the cells are migrating from one side to the other side, from the subpallian to the cortex. What will be in the real situation in vivo from the ganglionic eminence to the cortex. So if we just wait enough time, we can analyze how these cells migrate. So for that, we did time-lapse experiment after 30 days that the organoids were fused. So here, just to show you how this migration look like, sometimes the video at the beginning are a little bit slow, but with the time you will see that it's easier to see the migration. So for example, this is the control condition. You can see some neuron migrating over here, this one that is moving over here. So this one is the ARS condition. There are also many cells labeled and they are migrating. So you have an example here, here. So now what we did with these kind of movies is just follow each neuron over time. So you can see how in the control condition, the neurons are moving, the interneurons are moving, but in the ARS condition, they seem to migrate farther. So this is the track of every single neuron that we analyze. So you see, again, how the ARX are migrating farther. When we quantify the final distance, you see that in fact, they migrate longer distance, and also the velocity is a little bit faster. So for this first set of experiment, what we did was use subpallian and cortical control or subpallian and cortical ARX. So this is the same data that I just showed you, and this is the mean for all of these tracks. So you see how little by little these cells are going further. So now the next question was, is this phenotype is a cell autonomous effect? So in another way to say that, if the defect is a problem of the migration of the interneuron. So to try to tear apart where is the problem, so what we did now is fuse the subpallian from a patient and the cortex for a control. So now the interneuron will be mutant, but the context around will be controlled. So now what we did that, so you can see here that now the interneuron, they can migrate almost as good as the control did. So you see how now the distance that they migrate is reduced, and also the velocity is more similar to the control. So then we want also to study the other way around. What happens now if what is of the interneuron, the control, here in blue, and the cortex is the one that is mutated. So you can see here that now this control neuron migrate more similar to the mutation situation. It's not as bad, if I can say bad, like in the mutant condition, but you see that now they are migrating farther and also faster. So these results suggest that the problem of the interneuron migration is not due per se to the interneuron, or is not in the interneuron, is due to the defect in the cortex. So finally, the last experiment that we did, we wanted also to see does this defect in the migration also have consequence in the physiology of the neuron. So we did again with same MEA, and in this case, we fused the organoid more or less at the same time that we did the time-lapse recording. We did the MEA, and we divided the analysis in the fusion side, the cortical side, or the subpallian side. These results are really preliminary, but you can see that there is a slightly decrease in the activity in the subpallian side in the ARX patient. So in conclusion of the subpallian part, polyalanin expansion affects cell proliferation and increased cell death. And because the cortex is affected, now the cell migration of the interneuron migration is also affected. So finally, just the take-home message that I want to give you today is that ARS affect both cortical and subpallian development, and brain organoids are good model to study brain development and also neurodevelopmental disease. So our final goal is use this model in vitro. Hopefully we can use them for drug screening, and we can go back to the patient. So finally, I would like to thank all the people in the lab, Jenny to be my mentor and let me the opportunity to be in her lab, all the people in the lab, our collaborator, and thank you for your attention. So now I will be happy to take any questions. Thank you.
Thank you, Vanessa. That was great. So what I'm going to risk the bombers here and open up. If you have a question for Vanessa, you can unmute yourself and ask. It should work. I've locked the meeting, so I don't think anybody else can come in. So I'll ask a question at first. So, you know, you're growing your cells out to 120 days in vitro. How mature are all of your, different neuron types, your cortical neurons and your GABA neurons?
they are still immature.
They're still.
Because this is the problem with the organoid, that even if you maintain them longer, I did it also 180 organoids, but anyway, they are still immature. So for this reason, sometimes trying to see phenotype in electrophysiological phenotype is a little bit harder with the organoid.
I see. So can you get any type of sort of subclasses of neurons at all, or they're all just pretty immature?
So talking about at cellular level,
yeah,
in the cortex, yes, we can differentiate different layers. So like deep layer, upper layer, and even subtype of layer. So like layer 1, layer 2, and all of that. And in the subpallian, it's a little bit harder. So we can see some calretinine, calbindin, but they are expressing little by little. So not that much. So they express GABA, but for subtype of interneuron is hard.
I see.
I have a question. Vanessa, this is Laura. That was great talk. I was wondering about your migration result. So the organoids are larger, correct? The ones
Yes.
with the RX mutation. So I was just wondering if the further migration is actually abnormal, or is it just what you would expect? If they are going to the same target location, they have to travel further.
But if that is the case, when we are doing the mismatch experiment, that is how I call them, when we are fusing the ARX, for example, subpallian to the control organoid, that I don't think will be just because I just following the neuron like 15 hours. So it's not the whole migration path, the one that I follow, is just in that 15 hours. So I think in that way, it shouldn't matter that much the size of the organoid. And also, I was saying, in the mismatch, now is not exactly the same size. You know what I mean?
Yes, yeah. So do they eventually get to the correct position, or do you see them overshooting?
So I didn't follow them further. So now what I want to do is see after these 30 days, that is the longer that I wait just to let them migrate, see what type of cell they are migrating and what they have around. But it's hard in this case to see or to say what is the final location, because it's not like in the cortex that you know, okay, this neuron will go to layer 1 or to layer 2. So here is a little bit difficult to find the final destination because we are not sure what is the final destination over here. So what I want to see if at least if they tend to go more to one layer or to another, and if that is different in the ARX condition.
Thanks.
Thank you for your question.
Vanessa, this is Momoko. Thanks for the talk. So doing the organoid culture heavily, I know there are many variability in the organoid differentiation, and we specifically tried the Pascal protocol as well as Sasai protocol. And I found it's difficult to maintain the consistency in the organoid production in general. So I think it was great that you see the phenotype, but if you can comment on how you dealt with that kind of inconsistency and variability in the culture. And also recently Chris signed paper, they mentioned about the cellular stress, and then do you have any concern about that affecting the phenotype of the disease modeling?
Yeah, this is a great question. And as you say, we also experience the same kind of variability in the organoid. So for this reason, just to be sure that our phenotype is consistent, what we are doing is analyzing at least 2 clones of each iPSL line and 3 lines of control or patient. So in this way, only if the phenotype is maintained in all the conditions, we believe that the phenotype is real. So if it's in one line and not in another, if we cannot replicate enough the line that we have, then we don't trust that data. And also for this reason, also we are comparing immunostaining and single cell just to have different approaches to see if everything tells us the same. But you are right, the organoids are variable. So for this reason, the only way that we can control a little bit the variability is adding more organoids. So we are analyzed for each condition like 18, 20 organoids, just to also try to have a consistent phenotype. As you mentioned, at the beginning, we also was comparing different protocols, and we decided to use Pascal protocol because as you are adding factor, a little bit, you can have less heterogeneity. Anyway, they are still a little bit heterogeneous, but at least they are a little bit more consistent, at least when we compare with other protocols. And the second question that you say about the stress, that is also a possibility. Any kind of in vitro model produce an stress to the cell, and especially with organoid, all the people is talking if you produce or if it's hard as the organoid are getting bigger, is, for example, the nutrient cannot go to the core of the organoid, and that can produce not only stress, but also cell death. And at least in our organoid, because I did AC3, I didn't see many apoptosis cells in our organoid. We have some, and for example, in the subpallian, in the ARS condition, there are more apoptotic cells, but it's not that everything is dead or nothing like that.
Thanks.
Thank you.
Hi, this is Ben Novich from UCLA. Really interesting talk. I just had a couple of questions about the interpretations of some of your experiments and results. So I'm a little confused by the observations that the progenitors at one stage in the cortex are reduced in numbers, but then become increased in numbers. And I'm just curious, is there a mechanism or like any observations that have been seen from the mouse studies that would sort of support that's like what that effect is all about? And then the second question I had is, you are implicating that that it's a defect in the cortex that's affecting interneuron migration, and I'm curious, in light of what you're seeing in the cortical spheroids, how would that impact the migration? If there's a mechanism that you're thinking.
Mm-hmm. Yeah, this is both of them great question. So here is I just so here the summary of the cortex, just to try to explain what we believe is what is happening. So I think at the beginning, at 30 days, when the organoids are younger, so what ARX is doing is promoting the differentiation. So for this reason, at the beginning, there is a decrease in the progenitor pool because everything is just going through the differentiation. And because at the beginning, the neurons that are being forming are the deep layer neuron, this is why these deep layer neurons are increased now. So what happened later? So now could be that these cells are dying because they have some problem in the connectivity or something. So probably these one are dying. But now we have like another wave of neurogenesis that now the upper layer neuron is the one that are forming the most. So this is why now we have again like try to push the proliferation a little bit just to increase axis and also promoting the differentiation of the upper layer neuron. So I think this is why the phenotype is opposite if you compare younger versus older organoid. If we compare that with mice, the data that are in mice, I just saw one slide that, for example, when you knock out ARX just in the EMX1 population, so there they also found defect in the upper layer neuron. But I think also the problem could be that in mice, you cannot model completely this kind of layering defect because the cortex in human is expand. So maybe you can just slightly model this kind of defect in the mouse. So this is, I believe, what could be, or what is the limitation about the mouse model. Now you also ask about how this defect can affect the migration of the interneuron. So at least what our data is suggesting is that the interneuron can migrate, at least if the mutation is a polyalanin expansion. What is known in mice is that the knockout, the interneuron are stuck in the ganglionic eminence and they don't migrate to the cortex. However, in the polyalanin expansion model in mice, the cell can migrate. Sometimes they say that maybe there is a 80% of migration of neuron that migrate, but they can migrate. So what I think, the same reason that I was saying that sometimes you cannot model this kind of cortical layer defect in the mice because it's not expanded as much as the cortex, the human cortex, sorry. So I think also for this reason could be that in the polyalanin expansion model of ARX, you cannot see this kind of defect of migration defect of the interneuron, probably because the cortex is not as affected as happening in human. So that is my theory.
Okay, yeah, that's fine.
So I don't know if you have other suggestions that maybe can also be right.
Well, I mean, one of the things that I don't want to kind of get too deep into this, but like I'm still a little confused at like where exactly ARX is functioning in the context of the organoid, because I mean, like, is it expressed in the progenitor cells during like a phase of neurogenesis? Is it expressed in the nascent neurons? Is it expressed in the fully differentiated neurons? I think that there's many different interpretations that one could envisage. And I guess the thing that I'm just a little bit kind of concerned by is there's a lot going on in the morphological structure of the organoids is inconsistent. I mean, I would say that like there's and to try to judge layering in any organoid system is potentially problematic. But anyways, I'm just curious like how you're thinking about this and like what ways in which one might come kind of get around some of these these issues and questions.
Yeah, I think at least what the single cell RNA data show us is that ARS is expressed almost in every cell type that we check, both in the cortex and in the subpallium. So at the beginning, what is known in mice is that it's a key factor for interneuron, but I think it's more than that, because we can see expression is in almost all the cells. Maybe the level are different, and for sure the expression is higher in the subpallium spheroid in this case. But I think it's expressed in the progenitor, it's expressed in the neuron. So for this reason, we can find different, or we can find defect in more than one population.
Okay, thank you.
Thank you.
Any more questions? Going once, twice? Okay, well, Vanessa, I think you did a great job. Thank you for presenting. Next, we have a next seminar in October 1. It's going to be Lauren Lau from Kevin Staley's lab, and we will make a few edits to the link here to prevent Zoom bombers next time. But thank you, Vanessa. That was great.
Okay, thank you. Bye.